Hello everyone, this is Pratima and you are watching Planet Physiology. In previous sessions, we have studied classification of transport mechanisms and details about passive transport, mainly the diffusion. In this session, we shall study active transport across cell membrane. It is defined as transport of substances against the gradient by utilizing energy. Against the gradient means from low concentration of solute to high concentration of solute. It can also be against electrical or pressure gradient. Active transport requires help of carrier protein and metabolic energy. It is divided into three types: primary active, secondary active and vesicular transport. Some authors include vesicular transport under active transport, while others consider it as a third separate type of transport mechanism. But as many examiners expect students to write about vesicular transport under active mechanisms, I have included under active transport. So let us start with active transport processes. Primary active transport is defined as movement of substances against the gradient by direct utilization of metabolic energy. So here energy source is ATP. Important examples of primary active transport includes sodium-potassium pump, calcium pump, hydrogen-potassium pump and hydrogen pump. As these carriers utilize ATP for their activity, they have ATPase activity associated with them and hence they are also called as ATPases. For example, sodium-potassium pump is also called as sodium-potassium ATPase. Now let us study details of this sodium-potassium pump. It is present in each and every cell of the body. It was discovered in 1957 by Danish scientist Jan Christian Skov, who was awarded Nobel Prize for his work in 1997. Sodium-potassium pump is heterodimer made up of two protein subunits, alpha and beta. Alpha subunit has molecular weight of about 1 lakh and beta subunit has molecular weight of about 55,000. Alpha subunit has binding sites for 3 sodium ions and 1 ATP molecule on its intracellular fluid side. It also has phosphorylation site on intracellular fluid side. This pump has binding site for 2 potassium ions on its extracellular fluid side as well. It also has binding site for wabin on extracellular fluid side. Wabin is cardiac glycoside traditionally used as arrow poison in Africa. Beta subunit is a glycoprotein which attaches the alpha subunit to the cell membrane. Even though it does not participate in ion transport directly, it is must for normal functioning of sodium-potassium pump. Now let us study working mechanism of this pump with the help of simple animation. For convenience, only alpha subunit is shown here. These are the sodium binding sites and these are potassium binding sites and here is the ATP binding site. As you can note, this is extracellular fluid with higher concentration of sodium and intracellular fluid side with higher concentration of potassium ions. When 3 sodium ions and 1 ATP molecule bind to their respective sites on the pump, ATP is hydrolyzed to form ADP and inorganic phosphate. ADP is released but phosphate group remains attached to the pump, causing it to phosphorylate. This causes conformational change in the carrier protein and sodium ions are released into the extracellular fluid. Now, potassium ions bind to the binding sites and protein gets dephosphorylated. Dephosphorylation causes carrier protein to change back its original conformation and moves potassium to the intracellular fluid side. Okay, now let us study how this activity of sodium-potassium pump is regulated. Whenever there is swelling of the cell, it stimulates the pump automatically. Second messengers like cyclic AMP and diacylglycerol upregulate sodium-potassium pumps and hence the hormone acting via these two second messengers increase their number and hence the activity. Sodium-potassium pumps are inhibited by wabin and digitoxin. Digitoxin is used to strengthen the cardiac muscle contractility. It binds at the Wabin binding site. Okay, now what are the functions of sodium potassium pump? First, it maintains resting membrane potential. Let's study how. As we have seen in the mechanism, it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. Hence, the coupling ratio is 3:2. Both these ions are positively charged ions, and hence, every time this pump operates, it causes loss of one positive charge from the cell. Thus, it creates negativity inside the cell and contributes to -4 mV of resting membrane potential. As the pump generates electronegativity inside the cell, sodium potassium pump is also called as electrogenic pump. Next important function of sodium potassium pump is to maintain cell volume. Sodium ions tend to diffuse into the cell due to their natural gradient. As they are osmotically active particles, water also follows them. As a result, cell swells, and if it continues, cell can burst. But the prompt removal of sodium ions by sodium potassium pump prevents endosmosis and hence the cellular swelling. Thus, it plays important role in maintaining cell volume. If there is any accumulation of water into the cell, activity of sodium potassium pump is stimulated to remove this excess of sodium and hence the water. By pumping sodium into the extracellular fluid, sodium potassium pump also increases concentration gradient for sodium ions. This gradient facilitates transport of other ions along with them. Thus, sodium potassium pump acts as driving force for secondary active transport processes. Apart from these functions, sodium potassium pump also acts as signal transducer and helps in intracellular communication. For example, binding of Wabin leads to activation of SRC kinase. Another example is Wabin-induced mitochondrial reactive oxygen species production. Thus, due to their role in important cellular functions, sodium-potassium pumps are present in each and every cell of the body, and they consume about 30 to 70% of the cellular ATP produced. The next important example of primary active transport is calcium pump. It is also present in all the cells and in the membranes of cell organelles as well. These pumps remove calcium from cytoplasm and keep their intracellular fluid levels low. Calcium pump in L tubules of the sarcoplasmic reticulum play important role in muscle relaxation by removing calcium from sarcoplasm. Other examples are hydrogen potassium pump found in parietal cells of gastric glands and hydrogen pumps found in late distal tubules and cortical collecting tubules, cell organelles like lysosome, endoplasmic reticulum and mitochondria. This slide belongs to nice to know section. As I mentioned earlier, these pumps are also called as ATPases due to their ATPase enzyme activity. They are of 4 types. P-type, that is phosphorylation type ATPases, include sodium potassium ATPase, hydrogen potassium ATPase and calcium ATPase. These carriers work on the basis of phosphorylation and dephosphorylation as we have studied in sodium potassium pump. Second is V-type, that is vacuolar type ATPases and include hydrogen ATPase. The third is F-type, that is energy coupling factor type and includes mitochondrial ATP synthase. The last one is ABC transporters which include CFTR and MDR1 carriers. So here we finish the topic of primary active transport and now let us begin with the secondary active transport. Secondary active transport is defined as transport of substances against the gradient with the help of carrier protein by utilizing energy created by primary active processes. It is of 2 types, co-transport and counter-transport. First, let us study co-transport, which is also called as symport. In this case, sodium ions and the substance to be transported move in the same direction with the help of carrier protein. Note here that Sodium ions move along their concentration gradient created due to primary active transport. During this movement, sodium also carries another substance along with it inside the cell. As both the substances are transported in the same direction, it is known as co-transport or symport. Example of co-transport include sodium-glucose co-transport with the help of SGLT carrier protein. It is present in intestinal cells to absorb dietary glucose and in proximal convoluted tubules of kidney to reabsorb filtered glucose. Another example of co-transport is sodium amino acid co-transport found also in intestinal cells and proximal convoluted tubules. Note here that carrier protein can transport more than two substances also, and transport also can be from inside to outside of the cell, as in case of 1 sodium, 1 potassium and 2 chloride transport via NKCC transporter in loop of Henle. These ions leave the cell to enter the interstitial fluid. Let's understand symport with animation. As you can note, sodium gradient is from outside to inside, which is created by primary active pump. When sodium and glucose attach to SGLT carrier protein, it shows conformational change and both the molecules are transported into the cell. Thus, glucose is actively transported with the help of sodium ions. Next type of secondary active transport is counter-transport or antiport. In this case, sodium and the substance to be transported move in opposite direction. For example, in case of sodium hydrogen countertransport, sodium ions enter the cell and hydrogen ions leave the cell. It is present in proximal convoluted tubule. Another example is chloride bicarbonate countertransport, which occurs in RBCs. Here bicarbonate ions leave the cell and chloride enter the cell. Note that secondary active transport can involve ions other than sodium also. One more example is sodium-calcium exchanger in cardiac muscles. In counter-transport, as the substances are transported in opposite direction, the process is known as antiport or exchange. Let's understand with the help of animation. So, sodium and hydrogen bind to their respective binding sites on the carrier protein. The carrier undergoes conformational change and moves the sodium into the cell and hydrogen out of the cell. So here we have completed secondary active transport, and now the last type of transport is vesicular transport. Vesicular transport is meant for moving macromolecules like proteins which cannot be transported with the help of carrier protein also. It is not related to the concentration gradient and it may require energy in the form of ATP and calcium ions. It can be of two types, exocytosis and endocytosis. Exocytosis is transfer of molecules from cytoplasm to the exterior of the cell, and it requires calcium ions, ATP and docking proteins like V-snare and T-snare protein. V-snares are present on vesicle and T-snare proteins are present in the target location. These proteins guide vesicles to the specific locations for exocytosis. Exocytosis can take place either by non-constitutive or constitutive pathways. In case of non-constitutive pathway, protein from Golgi apparatus initially enters the secretory granules where they are processed further and then are released from the cell. For example, processing of prohormone to mature hormone before exocytosis. It is sometimes called as regulated pathway. Constitutive pathway involves prompt transport of proteins to the cell membrane for exocytosis. Also, the proteins in the vesicles show little or no processing or storage. Next type of vesicular transport is endocytosis, where cell engulfs the content. Based on the particles ingested and the regulatory pathway involved, it is of various types, which include phagocytosis, pinocytosis, and receptor-mediated endocytosis, which can be clathrin-mediated or caveoli-dependent endocytosis. Phagocytosis is also known as cell eating, where cell engulfs solid materials like bacteria or dead tissues. When the material makes contact with the cell membrane, it invaginates around it, and invagination is pinched off, leaving the engulfed material in the form of vacuole within the cell. The process of pinocytosis is same as that of endocytosis. Only difference is that engulfed material is liquid, and hence it is also called as cell drinking. Pinocytic vesicles are much smaller in size than that of phagocytic vesicles. Next is clathrin-mediated endocytosis. Clathrin is a protein with triskeleton state, that is, it has 3 legs radiating from central hub. Clathrin accumulates at membrane indentations. As the process of endocytosis begins, they form geometric array that surrounds the endocytic vesicle. As the vesicle is formed, a GTP binding protein called dynamin pinches off this vesicle from the cell membrane. Once the complete vesicle is formed, clathrin falls off and is recycled to form another vesicle. Clathrin-mediated endocytosis mainly responsible for internalization of receptors and ligands bound to them. For example, internalization of nerve growth factors and low-density lipoproteins. The last type is caveoli-dependent endocytosis. Caveoli are flask-shaped membrane depressions and literally means small caves. Accumulation of protein caveolin in the region of rafts lead to formation of caveoli at specific sites in the cell membrane. The sites of the cell membrane rich in cholesterol and sphingolipid are called as rafts. The substance to be internalized binds with the caveolin and the process is regulated by dynamin. Caveoli are involved in cholesterol regulation and transcytosis. They also limit movement of membrane proteins. Caveoli are prominent in endothelial cells where they help in uptake of nutrients from blood. Here is some nice to know section for vesicular transport. It is shown that vesicles involved in transport have protein coats around them. In humans, 53 coat complexes have been identified. Assembly protein 1 clathrin coat are present on vesicles that transport proteins from transgolgi to lysosomes, whereas assembly protein 2 clathrin coats are present on the endocytic vesicles that transport to endosomes. Vesicles that transport between endoplasmic reticulum and the Golgi, they have coat proteins 1 and 2. Also, there are certain amino acid sequences or attached group on the transported proteins which target the proteins and hence vesicles for the particular locations. RAF family proteins are small g proteins. They guide and facilitate orderly attachment of these vesicles. Humans have shown to have 60 RAB proteins and 35 snail protein types to regulate vesicular traffic. So let us wind up the topic by summarizing important points. Active transport moves substances against the gradient and hence require carrier proteins and energy. Primary active transport utilize ATP as energy. Important examples are sodium-potassium pump, calcium pump. Sodium-potassium pump plays important role in development of resting membrane potential and regulation of cell volume. Primary active transport in general creates driving force for secondary active transport processes. Secondary active transport use energy created by primary active transport. It is of two types, co-transport and counter-transport. In co-transport, two or more substances travel in the same direction. Example, sodium-glucose import. In counter-transport, two or more substances are transported in opposite direction and example is sodium-hydrogen antiport. Vesicular transport is for macromolecules and they may require ATP and calcium ions. It is of two types, endocytosis and exocytosis. Endocytosis can be phagocytosis, pinocytosis, clathrin-mediated or caveoli-mediated endocytosis. Exocytosis can be non-constitutive or constitutive. So here we finish with that active transport. Thank you for watching and see you in the next video. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.